Neural Lock NEET Series Day 3. Today we are going to see the cell envelope layers and the importance of each cell envelope layer. We are going to see about prokaryotic ribosomes and finally inclusion bodies reserve material. 3 blocks for today. So shall we start? Quickly grab your white sheets or notebook and pen and join with me. So first is the cell envelope layer. Very important question in NEET exams. It is seen in the prokaryotic cell, particularly bacterial cell. Cell envelope layer is a chemically complex layer. Chemically means the composition of the layers are different from outside to inside. See, NEET loves to scramble the order of arrangement from outside to inside or inside to outside. They will shuffle this and they will ask you. So today we are going to write it 5 times each in the order from outside to the inside. That is, this is the environment. You have outermost layer, then the middle layer, then the innermost layer, and this is the inner part of the cell. Okay, in this order we are going to write it 5 times, and so that we don't get panic during the time of exam. First is the outermost layer. You have to repeat it when you write it. That's how it will be retained in your long-term memory. So outermost layer is the glycocalyx layer, first layer. Second, middle layer is the cell wall. Innermost layer, plasma membrane. This plasma membrane is similar to that of a eukaryotic cell. Second time, shall we repeat? First layer is the glycocalyx layer, that is the outermost layer. Second, middle layer is the cell wall. Third layer is the plasma membrane, that is the innermost layer. Again, third time, outermost layer, glycocalyx. Middle layer, cell wall. The third and the innermost layer, plasma membrane. Fourth time, are you following me? Glycocalyx, outermost layer, middle layer is the cell wall, innermost layer is the plasma membrane. Then the final, fifth time, outermost layer is the glycocalyx layer, middle layer is the cell wall, and the innermost layer is the plasma membrane. Shall we revise it once more? Cell envelope layers, totally 3 layers seen in a prokaryotic cell. Here we are looking for a bacterial cell. It is a chemically complex layer, which means the composition of each layer is different and very important. From the outside to the inner side, 3 layers are arranged as outermost layer, middle layer, innermost layer. The outermost layer is the glycocalyx layer, middle layer is the cell wall, innermost layer is the plasma membrane. So how many of you have completed? If you have completed, leave a comment in the comment section. Then second is the importance of each layer. Okay, in NEET MCQ point of view, these are the code words which I am sharing with you. If you see glycocalyx, look for loose sheath and thick and tough. Loose sheath is a slime layer, thick and tough you call it as the capsule. That is, the glycocalyx is of 2 types. That is, it can be a loose sheath that is not attached firmly to the cell. You call it as a slime layer. If the glycocalyx is very thick and tough, you call it as a capsule. Shall we write it 5 times? We are going to write only this word, slime layer and capsule. These are the code keywords used in NEET examination. So, but we have to repeat this along with this, okay? If the glycocalyx is a loose sheet, it is called as slime layer. If the glycocalyx is a loose sheath, it is called as slime layer. Glycocalyx, loose sheath, slime layer. Glycocalyx, if it is a loose sheath not firmly attached to the cell, we call it as a slime layer. If the glycocalyx, that is the outermost layer, is very thick and tough, you call it as capsule. If the glycocalyx is thick and tough, it is called as capsule. The thick and tough glycocalyx layer is called as capsule. Thick and tough glycocalyx layer is called as capsule. Thick and tough glycocalyx layer is called as capsule. Now we saw the importance of glycocalyx in NEET exam. That is, if it is, they can ask you whether it is a slime layer or capsule, giving these words. Okay, loose sheath means slime layer, thick and tough means capsule. Next comes the cell wall. Okay, what is the importance of cell wall? Other than shape and structural support, it is very important on the basis of Gram staining. If it takes up the stain, you call it as Gram positive. If it doesn't take the stain, it is called as Gram negative. This is very thick. It has got thick walls. This one has got thin walls. Okay, thick walls. Remember this N. Thin walls. Okay, thick walls and thin walls. So if it takes up the stain, you call it as Gram positive. If it doesn't take up the stain, you call it as Gram negative. The one which picks the stain is called as Gram positive. Which picks up the stain is called as Gram positive. It is called as, it has got thick walls. It has got thick walls. Gram positive stain has thick walls. Gram positive stain has thick walls. Gram negative has got thin walls and it does not pick up the Gram stain. Gram negative does not stain and has thin walls. Gram negative does not stain and it has got thin walls. Gram negative does not pick up the stain and it has got thin walls. So the staining property is basically a characteristic of this cell wall. Out of 3 layers, only cell wall plays the role of staining. Okay, this one, only this is involved in Gram staining method. Rest of the 2 layers doesn't get involved. Okay, this is one important MCQ. Next is the plasma membrane. Like an eukaryotic cell, this is also selectively permeable, which means it acts as a gate. What has to enter inside the cell from the outer environment. It is selectively permeable, which means it allows only the specific one which does not disturb the inner part of the cell. Okay, it should maintain the homeostasis of the cell. So it cannot allow everything, right? So selective permeability is very important characteristic of plasma membrane. Plasma membrane is selectively permeable. They can confuse here. Selectively permeable, completely permeable, partially permeable. So all plasma membrane has the characteristic of selective permeability. Selectively permeable. Okay, first we saw the layers, 3 layers. Now the importance of 3 layers. The code words are for glycocalyx, loose sheath. Next is the plasma membrane. The word selectively sorry students, there is some network issue. Then block 2 is the prokaryotic ribosomes. Okay, in prokaryotic ribosomes, this one is important, this value, that is, it is a 70S architecture. Prokaryotic ribosomes has 70S and eukaryotic ribosomes has 80S. Okay, these are 2 subunits, that is, this 70S is made up of 50S and 30S. This is the larger subunit and this is the smaller subunit made up of 2 units. Okay, 50. This, see, I have marked it here. Sometimes we make silly in prokaryotic ribosomes. We have 70s type of architecture. If they give you 50s and 30s, don't add it. Okay, so it is a 50s and 30s is equal to 70s. This is a 70s type of architecture. So we are going to write 5 times. 70 is type of ribosome. 50s is the larger subunit. 30s is the smaller subunit. 70s is seen in prokaryotes. 70s ribosomes. 50 plus 30. 70s ribosome is made up of 2 units. 50s larger subunit and 30s smaller subunit. 70s ribosome type is seen in prokaryotes. 50s larger subunit and 30s smaller subunit. 70s type of ribosomes are prokaryotes made up of 50s larger subunit and 30s smaller subunit. Next, S denotes to, what does this S denote? It is called as Swedberg's unit. This denotes the sedimentation coefficient, which is an indirect measure of the density and size. I have written it in orders. This is repeated many times. S is the Swedberg's unit. It denotes the sedimentation coefficient, which is the indirect measure of density and size. So for you to easily remember, I have written it this way. S is Swedberg's units, which denotes sedimentation coefficient, is an indirect measure of density and size. These are the code words. You can directly look into the MCQ question. S denotes Swedberg's units. This is a sedimentation coefficient, which is an indirect measure of density and size. S denotes Swedberg's units, which is a sedimentation coefficient, an indirect measure of density and size. S means Swedberg's units, which denotes sedimentation coefficient, which is an indirect measure of density and size. S means Swedberg's unit, which denotes sedimentation coefficient, a direct measure in indirect measure of density and size. Okay, shall we go through once? Prokaryotic ribosomes are of 70s type. It is made up of 2 subunits, that is 50s a larger unit and 30s a smaller unit. S denotes Swedberg's units, which is a sedimentation coefficient, and this is an indirect measure of density and size. So you can remember these words and write it as many times as possible. If you forget, prokaryotic ribosomes, 70s, 50s, 30s, Swedberg's units, sedimentation coefficient, density and size. This alone you can remember. Okay, so next is the block 3. The examples we usually forget very easily. Under inclusion bodies, you have 3 examples. Okay, inclusion bodies are reserve material of prokaryotes. They do not have membrane, so they are openly in the cytoplasm. First is the phosphate granule, next is the cyanophyscian granules, next is the glycogen granules. Three granules. We are going to write the examples, each example 5 times, so that it gets locked in your memory. The three inclusion bodies of a prokaryotic cell: phosphate granules, cyanophyscian granules, and glycogen granules. Don't write this way, okay? Write like this, in this order. Automatically it will get registered in your subconscious mind. Phosphate granules, cyanophyscian granules, glycogen granules. Phosphate granules, cyanophyscian granules, and glycogen granules. Now you have a question. What are the two hormones involved in maintenance of blood glucose level? Name the two hormones which are involved in the maintenance of blood glucose level or blood sugar level. Leave the answer in the comment section. Phosphate granules, cyanophyscian granules, glycogen granules. Fifth time we are going to finish: phosphate granules, cyanophyscian granules, and glycogen granules. So shall we revise again once from the beginning? The first one we saw is the cell envelope layers. It is made up of 3 layers. It is a chemically complex structure. From the outside to the inside, you have outermost glycocalyx layer. Plasma membrane is selectively permeable. Next, what we studied, ribosomes. Okay, it is the prokaryotic ribosomes are of 70S type, made up of 50S and 30S, a larger subunit and a smaller subunit. S means Swedberg's units, which denotes sedimentation coefficient, and it is an indirect measure of density and size. Next is the final one, inclusion bodies. These are reserve materials of prokaryotic cell. It does not have any membrane, so it is openly in the cytoplasm. What are the 3 examples of inclusion bodies? Very important. It can be given in the match the following, like phosphate granules, vacuoles. Along with that, you have to pick the right match. So by heart it, always write down the examples as many times as possible. What are the 3 types of inclusion bodies? Phosphate granules, cyanophyscian granules, and glycogen granules. So students, tomorrow around 5:30 PM we'll meet again for the live class. If you want any particular chapter which you find it very difficult, you can type it in the comment section. I'll try to make a video out of that. Thank you for listening and following up.